kidneys produce about 180 liters of primary urine every 24 hours, but only 1 to 2 liters of urine is formed every 24 hours. 99% of the primary urine is reabsorbed by the uriniferous tubules. The uriniferous tubules include renal tubules and collecting ducts. Firstly, let's learn renal tubules. The renal tubule is lined by a simple epithelium. Its primary function is the selective reabsorption of water and other molecules from the primary urine. The renal tubule has 3 distinct histophysiological zones: proximal convoluted tubule, Renal's loop, and distal convoluted tubule. Renal's loop includes proximal straight tubule, thin segment, and distal straight tubule. We mainly focus on the proximal and distal convoluted tubule. Proximal convoluted tubule. Proximal convoluted tubule is lined by a single layer of pyramid-shaped cells with a well-developed brush border. The lumen is irregular. The cytoplasm is eosinophilic, rich in basal striations in the basal cytoplasm. The outline of the cell is obscure. The cell apex has abundant microvilli, which form a brush border, rich in mitochondria and membrane invaginations. The apical cytoplasm is rich in canaliculi, which increase the capacity to absorb micromolecules. Abundant lateral extensions interdigitate with neighboring cells. Functions. Proximal convoluted tubule is the initial and major site of reabsorption. About 85% of all ions, water, and all of the glucose, amino acids, and small proteins of the filtrate are reabsorbed. Secrete creatinine and substances foreign to the organism, such as hippuric acid and penicillin, from the interstitial plasma into the filtrate. Study of the rate of secretion of these substances is useful in the clinical evaluation of kidney functions. Renal's loop. Renal's loop is composed of the thick descending limb or proximal straight tubule, the thin limb or thin segment, and the thick ascending limb or distal straight tubule. Functions. Renal's loop can generate a hyperosmotic pressure in the extracellular fluid of the renal medulla. The thin limb is lined by a simple squamous epithelium, which has no capacity for active transport. These cells transport ions from the lumen into interstitium. As this part of the nephron has low permeability to water, the luminal fluid becomes hypotonic to blood. The vasa recta takes up water from the medullary interstitium and returns it to the general circulation. Distal convoluted tubule. Distal convoluted tubule is lined by a single layer of cuboidal-shaped cells with no brush border. The lumen is regular and larger. The cytoplasm is pale. The outline of the cell is distinct. The cell apex has few microvilli, rich in elaborate basal membrane invaginations, and associated mitochondria. the apical cytoplasm has no canaliculus. Functions of distal convoluted tubule: ion exchange, sodium is absorbed and potassium is secreted. The mechanism influences the total salt and water content of the body. Function is under the aldosterone regulations. Secrete hydrogen and ammonium ions into tubular urine. The activity is essential for maintenance of the acid-base balance in the blood. ADH can promote reabsorption of water in this section, concentrate urine, and reduce urine output. Collecting duct includes arched collecting tubule, straight collecting tubule, and papillary duct. Distal straight tubule is connected with arched collecting tubule, which opens to the straight collecting tubule. Straight collecting tubules drain into the papillary duct. In the right picture, the arrowhead shows the distal straight tubule. The cytoplasm of the epithelial cell is eosinophilic. The star indicates the straight collecting tubule. The epithelial cells are columnar and stained slightly. Collecting ducts concentrate urine by passive reabsorption of water into the medullary interstitium, following to osmotic gradient created by the counter-current multipedial system of the Renal loop. The amount of water reabsorbed is controlled by ADH, secreted by the hypothalamus in response to dehydration. They also can secrete hydrogen ion. Their functions are controlled by aldosterone, ADH, and atrial natriuretic factor. The blood passes from the glomerulus into the afferent arterioles, which at once branch again to form a peritubular capillary network that will nourish the proximal and the distal tubules and carry away absorbed ions and low molecular weight materials. They do not carry away the high osmotic gradient set up in the interstitium by Renal loop.